Hello, my name is Jaylynn Jones. I am an undergraduate research assistant in the DuCat lab. The project I have been working on this year is generating a bidirectional communication pathway between E. coli and cyanobacteria using quorum sensing. Native quorum sensing mechanisms use acyl-homoserine lactone, commonly known as AHL, molecules as a diffusible signal to induce responses in neighboring compatible cells. This allows for cell-to-cell communication in bacteria. The purpose of this project is to create a quorum sensing AHL signal between E. coli and cyanobacteria that is not originally there. Graduate students Mano Sinariz, along with myself, worked on this project by inducing E. coli with ATC and confirming that the E. coli could produce the AHL signal. Looking at the lower left figure, you can see that once cyanobacteria receives the AHLs, they can turn on the monomeric neon green, showcasing the AHLs have indeed been received. We then confirmed that cyanobacteria were able to receive the AHLs from E. coli. This is shown by increasing the neon green measured in cyanobacteria treated with AHL. As shown in the top left graph, the x-axis shows increasing AHL concentration with increasing IPTG levels. After we confirmed that E. coli produced AHLs and confirmed that cyanobacteria were able to receive the AHLs, we then added the sucrose permease CSCB and sucrose phosphatase synthase SPS genes under the AHL promoter. AHL turns on our two inserted genes, CSCB and SPS, which allows cyanobacteria to produce and export sucrose. We then performed a sucrose assay to measure how much sucrose cyanobacteria produced. We used two different concentrations of IPTG based on previous findings from Reese's heat map. The bar graph shows the millimolar concentration of sucrose produced by various IPTG and AHL concentrations. We were able to show how much sucrose was produced after 24 hours and found that there were the same range of production between higher and lower IPTG concentrations. We also showed that sucrose export is most dependent on AHL levels. We then were able to compare OD750 over some time while inducing with AHLs. This is depicted in the lower left graph. We found that at higher AHL, there was impaired growth. A take-home from this project is that we were able to successfully create a bidirectional communication pathway between E. coli and cyanobacteria using AHLs. This allowed the pairing of light harvesting and carbon fixing abilities of cyanobacteria with the biological production capacity of E. coli. Unfortunately, a limitation of this project is that these cultures are prone to contamination. However, the benefit of this project is that it is cost efficient due to us using cyanobacteria, which uses a self-inducing system. Cyanobacteria is also a carbon-fixing bacteria, so we have saved money on nutrients in our media. The future direction of this project is now that we have created a strain of cyanobacteria with AHL quorum sensing communication, it can be paired with other heterotrophs and used as a sustainable option for biotechnology companies. A huge thank you to my mentor Reese and my PI Danny for guiding me through this project.